Hello and welcome to Science Research Weekly, Season 4, Episode 6, Mantis Shrimps Punch Out. Get ready for a perfect Einstein ring, extracellular imaging adventures, into the candidate phyla radiation, Mantis Shrimps phonetic shields, InsectNet, and musings on laziness and CRAN in R. This episode covers materials from February 7th to February 13th. Let's get started with some research articles. First up here, we have Euclid, a complete Einstein ring, and NGC 6505. This is the Euclid spaceship. Was able to find a Einstein ring in a nearby elliptical galaxy, just about, I think, 600 million light years away, fairly close when it comes to the universe. And Einstein rings are artifacts or features of strong gravitational lensing, so they're very rare. So it was exciting to find them one so close, and I believe I have a better image of what that one looks like. So here's it far out, but then zooming in, you see this ring around the light source. Next up, live imaging of the extracellular matrix with a glycan-binding fluorophore. So cell biology, the main feature, of course, is cells, especially what's inside the cells, the intracellular components within a matrix. But it's easy to forget that, oh, right, cells are floating around an extracellular matrix. Now, it's actually hard to image that extracellular matrix, but researchers have been able to get this new fluorophore. So it's a small molecule fluorophore. It's cell impermeable, so stay on the outside. It's called Robo-6. And so it binds to glycans, and I think there's a lot of glycans in the extracellular matrix, so researchers can use this to really visualize the exterior of cells. So I think there's a bunch of looks like very high quality images. I'll just kind of click on one. So, okay. So yeah, a lot going on there. So that's exciting. And really a paradigm shift of thinking, oh right, yeah, the extracellular matrix is big and important too, not just the intercellular. Anyways, we'll switch gears here to our next article. Now, I'm going to break ranks and not give the normal precedent of the full name of the article because it's incredibly unwieldy, so I'll just shorten it to Minisyncocccus archaeiphilus gen nov species nov a mesophilic obligate parasitic bacterium. And it goes on like that to do family, order, class, phyla within the Candidatus Patescibacteria. So anyways, one of my favorite, in fact, my favorite candidate bacteria phylum is Patescibacteria, also known as the candidate phyla radiation, or CPR, or microbial dark matter that's sometimes thrown around. It's this huge swath of poorly characterized, uncultured microbes found in the environment, bacteria found in the environment. A lot of them are very small. A lot of them are not free living. Anyways, and so this new species, Minisyncoccaceae archaeiphilus, is the type species of an entire new phylum that they're proposing, and it's the first one that parasitizes an archaean. So archaea is the other branch of prokaryotes. So that's really interesting. I always like to see this sort of microbial, the microbial frontiers and how we get there. All right, next up here, I'm gonna have to shift this because it's hard to see with the screen. All right, the next article is, does the mantis shrimp pack a phononic shield? Phononic shield. So, mantis shrimps, aka the most charismatic arthropods of the ocean, aka stomapods. They come in a couple different forms. There's sort of the raptorial ones with stabbers for their arms, more like an actual mantis. And then there's the smashers, which essentially have like bowling balls or punching bags for arms. And they can actually punch so fast that they create cavitation bubbles that collapse and like send shock waves, so they can like break open muscles and other invertebrates. But the question is like, well, why don't they get hurt by their own punch, essentially the force from their punch? Well, using various, I think, high tech systems, researchers were able to detect that they can essentially dampen the waves coming at them, sort of shielding themselves from the worst of the effects. They use all sorts of things like blotch harmonics, flat dispersion branches, ultrasonic wave modes, Bragg band gaps. I'm sure it's all very complicated, but I think that the summary point is, yeah, they do have a shield that shields them from the effects of their insane punches. All right, moving along here. Ocean stratification impedes particulate transport to the plumes of Enceladus. So Enceladus is one of the moons of Saturn. It is a potential source of extraterrestrial life because it's possible there's a liquid ocean under its icy shell. However, we may never know that. Why? Well, water vapor plumes can erupt from the South Pole. However, based on some modeling of like ocean dynamics, the idea is that particulate matter, like organic matter, that may be prominent at the bottom. It might take centuries, depending on how the ocean is set up. It might take centuries for it to get from the bottom, the hydrothermal zone, to the surface. So those organic molecules will be degraded beyond what a probe that we'd send could detect. And so there's, I think, some pretty interesting. I think Figure 1 kind of shows it here. Now, if sort of there's unstratified convection, sure, you could probably get there, but if they're inverse stratification, it's really going to dampen it down, so it gets, yeah, on the order of centuries for things to move up the layers. All right, next article here. InsectNet, real-time identification of insects using an end-to-end machine learning pipeline. This is sort of like, yeah, machine learning, like an end-to-end software. And its real goal is to help, especially in the agricultural field, identify insects by photographs. Both, different stages of developments, confusing morphs, camouflage, things like that against challenging backgrounds. So hopefully helps solve those sort of problems. So yeah, it's very interesting. Obviously, there's a conceptual. It's already, I think, had fairly high success rate at correctly identifying. It starts with a database. Here's some more for the video watchers. There's some images of how it kind of detects. It can give scientific name, common name, other possible predictors. So yeah, it's really, really, I think, interesting and hopefully a game changer for application of insect detections. So people can detect, predators, pests, potentially dangerous species, etc. So hopefully that keeps on moving along. All right, our final article for today is surface lipids in nematodes are influenced by development and species-specific adaptations. So using this high-tech thing called 3D Orbi-SIMS, which is an advanced surface-sensitive mass spectrometry method, researchers were able to actually characterize the cuticle, the outer regions of nematodes, both species C. elegans and Minisyncoccus pacificus. It turns out that most of the surfaces are dominated by lipids, but the actual specific composition kind of can change across species and also developmental period. So I think that was really interesting, the idea of, yeah, think about spectrometry of like surfaces, living surfaces. So very, very cool thing there. All right, let's switch to some from articles to tools. The 2025, February 2025 Julia Hub newsletter is available. you're a Julia fan, with all sorts of sections of podcasts, webinars, news, etc. The analysis factor has a new post, the best reasons to run a pilot study, whether it's sample size, logistics, or problem solving. A pilot study might be a good idea. Let's turn our attention to our bloggers' posts. There's 4 that I thought might be useful. First, from our tutorials archive, Statistical Aid to School of Statistics. It's two-way repeated measures, ANOVA and R. So it seemed like a pretty straightforward and exhaustive, or at least expansive view of the topic in R. Sometimes repeated measures ANOVA can be a little trickier, so that might be a helpful resource. Next up, Lazy Introduction to Laziness in R by RHub. So it goes into the idea of what does it mean by lazy in R code, whether it's evaluation, lazy database operations, or lazy loading, and other terms. So kind of laying out what that actually means. Next, there is the article, Is CRAN Holding R Back? By Ari Lamstein. The idea about CRAN, that's the sort of major loader of repositories or packages. Maybe that's the way you could put it. Some of the issues with that is sometimes when a package will be archived from CRAN, and then other packages dependent will be archived, and the reason for it to be archived is sort of minimal, and so that can have a big impact on the R ecosystem. I think there's other topics in there as well, but that's one of the main points. So there's that. And then finally, estimating a Bayesian proportional hazards model with from R data generation by Keith Goldfeld. So it's, using more advanced survival analysis in R, which is something I'm kind of interested in. Maybe you would be too. All right, with that, we also want to talk about some new, I think I can stop sharing. Some new R bloggers posts, since we already talked about that. There is r.alpha.a.home, feel at home using R thanks to shortcut functions, making it simple. There's power HE, power and sample size calculations with hierarchical endpoints, and MIC, analysis of antimicrobial minimum inhibitory concentration data. That closes out our tools here. For research funding, there's no new grants, and there probably won't be for a while, so get used to it. With that, if you have any interesting recent scientific papers or tools you think I'd like, leave a comment on YouTube or Spotify, wherever you're listening or watching from. And do all that like and subscribe stuff. I want to bring more science goodness to the world. Stomapod, hexapod, Udapod. Thanks for joining me for Science Research Weekly. Science on.